The mathematical improbability debate. The mathematical challenge of calculating the probability of life's emergence has fascinated scientists and philosophers for generations. Now, with our growing understanding of quantum biology, this calculation has become significantly more complex. When we try to determine the odds of quantum-capable DNA forming by chance, we enter mathematical territory that stretches the limits of probability theory. Traditional biological structures are already remarkably intricate. A single human cell contains approximately 30,000 different proteins, each folded into precise three-dimensional shapes necessary for their function. DNA itself consists of billions of nucleotide bases arranged in a specific sequence. The probability of such complex structures forming randomly has long been debated in evolutionary biology. Quantum biology adds additional layers of complexity to these calculations. The physical arrangement of atoms and molecules matters critically alongside their quantum mechanical properties. For DNA to function as a quantum system, it requires precise arrangements of electron clouds, specific distances between atoms that enable tunneling, and molecular structures that can maintain quantum coherence. These quantum requirements significantly constrain the possible arrangements that would result in functional biological systems. Consider finding a specific grain of sand on all the beaches of the world. This gives us a glimpse of the probabilities involved in forming complex biological structures randomly. Several mathematical models have been used to calculate these odds. The simplest approach multiplies the individual probabilities of each component forming correctly. For a moderate-sized protein of 300 amino acids, with 20 possible amino acids at each position, this calculation yields a probability of 1 in 20 to the power of 300, an astronomically small number. When quantum requirements enter the equation, the probabilities shrink further. Quantum tunneling in enzymes requires atoms positioned at precise distances and angles. Maintaining quantum coherence demands molecular structures that shield quantum states from environmental decoherence. These constraints dramatically reduce the mathematical probability of such systems forming by chance alone. This mathematical territory has become central to debates about life's origins. Proponents of intelligent design highlight these calculations as evidence for their perspective. They point to probabilities like 1 in 10 to the power of 183 for specific protein sequences forming randomly, concluding that such improbability points to deliberate design rather than chance. However, evolutionary biologists offer substantial counterarguments that transform the probability landscape. Natural selection fundamentally changes these calculations. While mutations occur randomly, selection pressures consistently favor certain outcomes, making evolution a non-random process. Instead of requiring all components to form simultaneously by chance, evolution works through incremental steps, each providing some survival advantage. The vast time frames available for evolutionary processes also alter probability assessments. Life's 3.8 billion year history on Earth provides enormous time for evolutionary exploration. With trillions of organisms existing simultaneously, each potentially experiencing mutations, the probability space has been extensively sampled. What seems improbable in human timeframes becomes more reasonable over geological time. Selection pressure makes improbable structures achievable through natural processes. Even small improvements in function can provide survival advantages. Once a basic function emerges, selection can rapidly refine it. This directed optimization means evolution doesn't blindly search the entire possibility space. It follows paths of increasing fitness, significantly improving the odds of complex structures forming. Computer simulations confirm this, demonstrating that when selection is applied after each random step, complex structures reliably develop from simple starting conditions. These models suggest quantum-capable biological systems could evolve through similar processes of incremental improvement under selection pressure. A critical error in probability calculations involves what researchers call the post hoc probability fallacy. This occurs when calculating the probability of a specific outcome after it has already been observed, like calculating the odds of a specific protein sequence we see today. This approach ignores that many different sequences could fulfill the same function, and evolution had no predetermined target. As research notes, In most cases, the post hoc fallacy completely nullifies the argument. The relevant question isn't what are the odds of this exact arrangement forming, but what are the odds of some functional arrangement forming? Genetic redundancy and duplication further enhance evolutionary probability. Many biological structures evolved from simpler precursors through gene duplication followed by specialization. This pathway allows complex systems to develop without requiring simultaneous random formation of all components.